afternoon, everyone. Thanks for listening to my presentation. So my topic is about the detection of the foodborne bacteria. So let's talk about some bacteria today. Have you ever experienced foodborne poisoning? I hope not, because it's not fun. Everyone knows. It may include the major symptom is diarrhea, but maybe also like a vomiting, nausea, or fever. It's really not fun. And let me show you some numbers. So there are about 1 in 10 people feeling ill every year all over the world. And this caused more than 420,000 people die. And also it costs a lot of money. So the common foodborne pathogens, you may heard it or not, include like listeria, salmonella, E. coli, those three you may be familiar, and then Campylobacter, staphylococcus. So those bacteria, they are very easily to be transferred to our human beings because they exist almost everywhere, like in the environment, the water, and the animals, also all different types of the food. So they can be easily transferred to us, and it's closely related to our daily life. You cannot get rid of them. So there are already many different types of methods that have been used to detect those foodborne pathogens, like put them on the agar or use some sophisticated equipment to detect their DNA, their proteins, or other stuff. However, most of these methods, they have some drawbacks. They either very time-consuming, like labor-intensive, and they may require very highly trained personnel. Also some very expensive reagents. In that case, we need a better methods to detect them. So Raman spectroscopy is a good candidate. What does it work? So the laser hits on the sample. Then the Raman signal can be generated and collected within several seconds. And those signals are related to the vibrational modes of the functional groups in the molecules in the samples. So they are very specific. And when we test it, it's very easy to use. We just put the sample under the microscope. Then the single cell can be trapped by the laser. And then the Raman spectra can be generated in several seconds. And those spectra are like the fingerprinting of the bacterial cells. So different bacteria, they have different fingerprintings. And based on this spectra, we can know which bacteria is which. So here comes my study. My study is to use the single-cell Raman spectroscopy to identify or differentiate different foodborne bacterial pathogens, and also with the help of machine learning. So how I do that? Firstly, I grow all the different types of foodborne bacteria in the lab, and then collect them. And then I will put those bacterial cells under the microscope. So here is a schematic figure. This represents the bacterial cells trapped by the laser, and then the Raman signal can be generated. And here is the Raman spectra we collected from those different bacterial cells. And then, so this one looks complicated. So these signals, we cannot differentiate by using our naked eyes. Then here comes the machine learning. So machine learning is kind of very powerful. They can almost do most of the jobs for us. And this is the structure of my model. So after well training, it can tell us which one has the highest score. So those is the bacteria species that predicted by the model. So we can know the name of the bacteria we are testing. So let me show you some results here. As we can see here, different bacteria, they have unique Raman spectral patterns as indicated by different color. So they have different peaks at different intensities, but there are also some minor differences between those spectra as indicated in this area. So those teeny tiny difference, we cannot use our eyes to differentiate. We use the machine learning to notice this difference and then identify the different bacteria. So here is my final results. Looks complicated, huh? But you don't need to know the details. What you need to know is by using our methods, we can achieve an overall accuracy of over 95%. So what does this mean? Which means you're using our model, you're using our equipment and the machine learning model, then you have a very high chance, let's say it's over 95%, that you know what's the name of this bacteria that you want to know. So this is the final goal of my project. So in summary, my study, we developed a novel methods of using single-cell Raman spectroscopy and machine learning to detect different foodborne pathogens. And our methods is rapid. For collecting one spectra, it only takes like several seconds, and also it's reliable. The accuracy is over 95%. And most importantly, it's very easy to use. I believe after some basic training, most of us in this room, we can detect them. We can use these methods. So because all these advantages, this method has a high potential to be used for different places, like in the food industry, in the government, or even in the hospital. So people can use it to identify different bacteria. Yeah, finally, I'd like to acknowledge my supervisor, all the lab members, and my funding agencies. Thanks for your attention. Any questions?